Hello YouTube. All right, so all we did was just go over ATP and acetyl-CoA. Everything they need to know is in these in the word, right? In the word of the PowerPoint. But yeah, like really my PowerPoints is just me facilitating the information, making sure it comes through. So we went over ATP, acetyl-CoA, NADH, FADH2, insulin, glucagon. We were talking about how the inhibition of certain enzymes occur in pathways. That the main ones that you should know, there's a multitude that I sent to you guys, but the ones that you should really ought to know is the influence of ATP and AMP on the cell, and also the influence of insulin and glucagon on pathways. Yeah. Good. One pepper. Thank you. All right. So we can get started with the Krebs cycle. All right. So in the Krebs cycle, and the way I like to memorize insulin and glucagon is like the disease of diabetes. Like it makes me like first it's medicine, and then so it's interesting, and then you have the insulin glucagon there. Okay. And you guys remember, if you ever have any questions about like these pathways or stuff, definitely reach out to me. All right. So next we have the Krebs cycle. So in the Krebs cycle, it happens in the mitochondrial matrix. So we're transferring from the glycolysis, which was in the cytoplasm, to PDC, which took the pyruvate from the cytoplasm and entered it into the mitochondrial matrix. And the Krebs cycle is also in the mitochondrial matrix. Remember, the mitochondria looks like this: an outer membrane, the inner membrane, which is called cristae, and then you have your intermembrane space. So the outer membrane, this is cristae, and then you have your matrix here. Right. So the Krebs cycle happens in the matrix, the nucleus, or the DNA of mitochondria. Mitochondria also have their own DNA. Is also in the nucleus. The ribosomes are also in the nucleus. Sorry, in the nucleic acids and ribosomes of mitochondria are also in the matrix. Okay, so basically what the Krebs cycle does is that it takes the acetyl-CoA that we produce from our pyruvate dehydrogenase complex and just oxidizes them to oblivion, right? So like here in our acetyl-CoA group, for example, we start off with like some hydrogens here, they're cool. But by the end, we just get carbon dioxide, right? Which is 0 hydrogens, is super oxidized, right? So we're going to take a bunch of electrons from our acetyl-CoA. So in the Krebs cycle, what happens is that the acetyl-CoA is introduced into a compound called oxaloacetate. That oxaloacetate is going to be not decarboxylated. So like the two carbon dioxide groups that you find that are produced from the Krebs cycle, they're purely from the acetyl-CoA, right? Like the acetyl-CoAs go from so there's 2 carbons in the acetyl-CoA, the carbonyl carbon and the alpha carbon. Those 2 are the ones that are sent out as CO2 molecules. The precursors of the Krebs cycle, like the intermediates that are there, they're not going to be decarboxylated. They're just going to shuttle the acetyl-CoA through the oxidation steps. Okay, so as we mentioned before, the glycolysis is an anaerobic reaction, meaning that it doesn't need oxygen. The Krebs cycle is a oxidative pathway. It doesn't necessarily need oxygen for it to occur. The reason why it's an oxidative pathway is that ETC, which is the pathway after the Krebs cycle, requires oxygen. So in a way, indirectly, the Krebs cycle needs oxygen for it to occur, right? Because if there's just a buildup of product, right, the product of the Krebs cycle we mentioned was the NADH and the FADHs. Well, if like we didn't have any oxygen, then the ETC wouldn't be running, and then we would have a buildup of NADHs and FADHs. So the Krebs cycle would just stop, and that's why it indirectly needs oxygen, the Krebs cycle, in order for it to run. Okay, so the Krebs cycle, along with it, what's it called, being inside the mitochondrial matrix, it's shielded from hormonal control, right? Like things like insulin, things like glucagon are not going to be affecting the Krebs cycle. The only thing that affects the Krebs cycle is the actual intermediates or reactants that are found within the Krebs cycles. Like NADH is going to affect it, ATP is going to affect it, but nothing hormonal like the insulin or glucagon, because the mitochondria shields it from entering into the Krebs cycle. Okay, so next is just some bits of information. So whenever ATP is high, like if you're, let's say you just ate and there's like a bunch of ATP produced from glycolysis and ETC, and so the cell is like full of ATP. So when the concentration of ATP is high, the citrate is going to shuttle the acetyl-CoA into the cytoplasm for fatty acid oxidation. So that's basically the Krebs cycle trying to be as efficient as possible, because if the Krebs cycle just kept going, then you just have a bunch of ATP that you have nothing to do with, right? So instead, it's going to store that potential energy. Remember, the acetyl-CoA is this group right here. It's going to store that potential energy into a fatty acid. It's going to take those two carbon molecules, like this carbon molecule here, this carbon molecule here, and it's going to create a chain, and that chain eventually becomes a fatty acid, and that fatty acid is stored. And that's why they say that sugars can become fats, but fats can't become sugars. The reason why sugars can become fats is because you can get a glucose molecule and can turn into an acetyl-CoA, and if you need to store it, you can store it as fat. However, remember the PDC, the pyruvate dehydrogenase complex, that complex that we did in yesterday's class, that's an irreversible complex, right? So you're not going to make, what's it called, acetyl-CoA. Remember, the acetyl-CoA was the product of the PDC. You're not going to have the acetyl-CoA turn back into the pyruvate in order for that pyruvate to go into like gluconeogenesis, something I'm going to be discussing in a recorded lecture. And gluconeogenesis is just the production of glucose from intermediates. So that's why sugars can turn into fats, but fats can't turn into sugars. Okay. Because when you oxidize a fat, you just make acetyl groups. You just make more acetyl-CoAs, and those acetyl-CoAs can't turn into something that can be produced. Can't turn to something that produces glucose. So next, so that's what happens when ATP is high. However, when ATP is low, or you are in a starved state, then what you can do is some amino acids are going to give up their carbon backbone. So amino acids do have carbon backbones, and they're going to give it to some intermediates that are found in the Krebs cycle. Like for an example, an alpha-ketoglutarate. Right, glutarate is one of the amino acids that you're going to be learning tomorrow. This amino acid can go into the Krebs cycle and help it out in case like there's not enough intermediates in there. Okay, so the products of the Krebs cycle is 6 NADHs, 2 FADH2s, 2 GTPs, and 2 CO2s, right? So another name for the Krebs cycle is the citric acid cycle. We'll see why in a second. Also the tricarboxylic acid cycle. Again, here's just some like bits of information that I collected. Okay, so here's the mechanism. Okay. So I have the structures and the enzymes written down. I'm going to briefly just run through the Krebs cycle. You guys should be able to recognize how the enzymes are pretty much conveniently named, and how the products of a reaction are also very conveniently named. So here we have succinate. I would say there's no easy way to like. I mean, I'm sure someone can create a metonomic for why succinate looks like this, but I just think of it as just like kind of like a U shape, like this. Like this is succinate, and the succinate we have a U. That's the only way that I can think of to remember it. So this is here in total 4 carbons, right? 1, 2, 3, 4, and we have a COO. That just means that we have a carboxylic acid, right? It's a carbon. It's a carbon with 2 oxygens and a hydrogen. So a COO just means a carboxylic acid, okay? So here we have a carbon with a carboxylic acid. Here we have another carbon with a carboxylic acid. That's a succinate. We're going to be using succinate dehydrogenase. So the dehydrogenase is a redox reaction. In this case, we're going to be using FAD. And whenever we have FAD, we're going to be working with carbon-carbon bonds. So remember before with the NADH, and we were talking about the glycolysis pathway, the NADHs were affecting the alcohol groups, but in this case for the FAD, we can be interacting with the carbon-carbon groups. And since this is FAD going from FAD to FADHs, that just means that the H's from the succinate was taken off and placed on the FAD. Can anyone tell me if this is a oxidation or a reduction?
Oxidation.
Yeah, it's an oxidation because the hydrogens were kind of plucked off the carbon. Okay, so next we have fumarase, which is just fumarate and then just A's at the end. So this is fumarate. That's the product of succinate dehydrogenase. Yeah, fumarase. You introduce a water molecule and you have an L malate. If the water molecule was like if the OH group was pointing that way, you would have a D malate. If it's here on EL, like L left. So here you have an L-malate dehydrogenase, and then we have this time a NAD+. So I'm going to be working with the alcohol group, and you're going to make a carbonyl to make oxaloacetate. This is a really, really important structure for you to know. Oxaloacetate is found in a lot of pathways. Okay, oxaloacetate, usually abbreviated as OAA, is going to turn. So you guys don't have to worry about like copying this down because like they're always going to be in the slides. Like no reason to like rush and copy it down right now. So just more listen to the explanation. So here we have oxaloacetate. The alcohol group from the L-malate has been oxidized, right, to a carbonyl group. Like the hydrogen was taken off. Now we have a double bond to the C. And I want to introduce our acetyl-CoA that we got from the PDC, right? So this is where the acetyl-CoA is going to interact with the Krebs cycle. So the acetyl-CoA with citrate lyase is going to combine the oxaloacetate with the acetyl-CoA, and then briefly make something called the cityl-CoA, where we can see here we have 6 carbons. Remember, the acetyl-CoA has 2 carbons. The oxaloacetate has 4 carbons. Here we have our oxaloacetate in this group right here. So it's 1 carbon, 2 carbon, 3 carbons, 4 carbons. Just the carbonyl that was on the ketone right here, that was on the oxaloacetate, has been reduced. The reason being is because you can't what's it called? The acetyl-CoA is going to be attaching to this carbon right here. Right? This is the acetyl-CoA. This is 1, 2, and you can't have a double bond on this carbon if you're having another bond come in. So the carbonyl group gets a hydrogen. You have a cityl-CoA when you have this structure. The CoA comes off. You have a water molecule come through, and then you have citrate. And this is also why it's called the citrate cycle, the citric acid cycle, the CAC, because this is the citrate molecule, also known as the tricarboxylic acid cycle, right? There's 1, 2, 3 carboxylic acids. Next, we have aconitase, which is going to be like pretty much an isomerase. So aconitase plucks off a water molecule. We get a bit of an oxidation, and go from having hydrogens here to not having hydrogens. And the aconitase just puts it somewhere else, right? So you go from citrate to isocitrate, right? Kind of like an isomerase of citrate. So it's aptly named. It's awesome. So citrate here, and the water molecules are the OH group, instead of being in the middle, it's here at the end, making an isocitrate. Did someone have a question? Okay, if not, we take our isocitrate, we use isocitrate dehydrogenase, again, aptly named. We're going to, oxidize our group. Right here, we're going to be oxidizing this carboxylic acid. Remember, a carboxylic acid can turn into a, into a CO2 molecule, right? This is what a carboxylic acid looks like. All you would have to do is just combine another bond here to this oxygen. This hydrogen gets plucked off, and then you have your CO2. So this carboxylic acid is going to be removed, right? Because the isocitrate dehydrogenase uses an NAD+ to oxidize our isocitrate. Briefly, we get a oxalosuccinate, and the CO2 comes off. And then we have an alpha-ketoglutarate, which we mentioned before, that amino acids can enter in order to help the Krebs cycle. Next, you have an alpha-ketoglutarate dehydrogenase. Same thing is going to happen. You're going to oxidize your molecule. A CO2 is going to be released. You're going to also combine a CoA enzyme, going to be attaching to the this here group. This is really that. And then from here, the succinyl-CoA synthase is just going to remove the coenzyme and also produce an ATP molecule. So this is one round of your Krebs cycle. And remember, because we produce 2 acetyl-CoAs from the PDC, because we have 2 pyruvate, and if you oxidize 2 pyruvate, you get 2 acetyl-CoAs. That means we're going to get this cycle twice. And that's why everything here, the products, is 6 NADHs, because we in one round we made 3 NADHs. In one round we made 1 NADH, and one round we made 1 GTP. So this ATP here at the end is a GTP, but for the tense of the MCAT, the GTP and the ADP and the ATP, pretty much the same thing. And then you get this should be 4 CO2s. Okay, any questions on the Krebs cycle? That's basically, that's all you need to know for the Krebs cycle. Wow. Okay. Any questions? All right, if not, again, yeah.
Sorry, how in depth do you think we have to know the structures of each, like, intermediate?
I would say you don't have to, but again, it's really helps with memorization, like knowing what this looks like. Okay, and then a dehydrogenase, if you're using an FADH, if you're using an FAD, you're gonna have this double bond here. So I would just, you don't have to memorize the structures. I'll just say memorize the names and the enzymes and like what the enzymes use. The structures are, least important, but at the same time, they really facilitate like the understanding of it.
Okay, thanks.
So the oxaloacetate can be recycled. It can be, oxaloacetate can go into gluconeogenesis, something we talked about, that I'm going to be talking about in the recorded lecture. But also, again, as you can see, the succinate is produced at the end, right? And so the succinate is just going to go back and make the oxaloacetate, right? So the only things that really happen here in the Krebs cycle is that the acetyl-CoA combines with the oxaloacetate and gets super duper oxidized and then leaves as a CO2. So this cycle just keeps going. However, the oxaloacetate and some of these other molecules like citrate and almalate, they're also used for other pathways. So sometimes they get snatched out of the Krebs cycle. So sometimes you're going to have to produce some intermediates in order to keep this cycle going. And then here we have the inhibitors. So I really just remember the NADHs, the ATPs. Everything else can be like, I would say, honestly, I would say like for the Krebs cycle, I would say like know all these inhibitors, for being honest. And most of them is just like kind of, it makes sense. Like if you have isocitrate dehydrogenase, for example, right here, right? What is it inhibited by? NADH. Okay, well, that makes sense because that's a product. So it's going to kind of inhibit it, like a negative feedback. And then ATP, right? There's a lot of ATP in your cell, then you're not going to want to do Krebs cycle. So a lot of these inhibitors are like it takes a single step for it to like click. Okay, any questions about Krebs before we move on to ETC? All right, so if not, ETC, the electron transport chain, is in the mitochondrial cristae. So you mentioned that the mitochondrial cristae, which is this, here's like a second membrane. This would be the first, the outer membrane. That would be the inner membrane. It's called cristae. So what the ETC does is it takes those NADHs and all those FADHs that we produced in the Krebs cycle, the ones that we produced in the pyruvate dehydrogenase complex, and the ones that we produced in the glycolysis. It takes all of them and kind of just takes their electrons, remember their electron carriers. It takes those electrons, it shuttles them through some complexes, and with that energy, with the negative energy of the electrons, because they're negatively charged, the proton is going to be pumped out, and that proton gradient that's existing above the proton gradient would be here, the intermembrane space, and then the protons, since like they're they have higher concentration in the intermembrane space than they do in the matrix, they're going to go down the gradient, and we're going to use that force that those protons use, that those protons happen to have when they're coming back down, to churn a enzyme that produces ATP. Okay, so again, the goals for ETC is just to regenerate. Oh, okay, sorry, I didn't say that. But again, like you're going to need ADP. You're going to need AD plus, NAD plus. You're going to need FAD in order to allow these other cycles to keep happening. Because, for example, here you need ADP. So you need AD for this enzyme to work. You're going to need NAD plus for this enzyme to work. And so in order to get those pluses back, you're going to make sure that those electron carriers empty their electrons into ETC, and then you can get those electron carriers back in order for these processes to continue going. So that's one of the purposes for the ETC. The second purpose, and probably the primary purpose, would be to produce the ATP, right? Okay, so the electron transport chain, your NADH and your FADH are going to be producing the proton gradient, as we mentioned before. And the enzyme that produces the actual ATP, we're going to have something called the Racker's knob. And what the Racker's knob basically does is that it turns and turns and turns while the protons are being shuttled out. Like, so the protons are like making this enzyme turn. And while it's turning, it's going to take an ADP and a phosphate and kind of like squeeze them together to make an ATP. That's how the ETC produces ATP through oxidative phosphorylation. Right? We had substrate level phosphorylation, for example, over here, substrate level phosphorylation, right? the molecule donates a phosphate group. But in ATC, we're going to have an oxidative phosphorylation, where the oxidation of the electron carriers, like you're oxidizing these electron carriers to get their electrons out, is going to be the process of ATP production. So just to quickly go over, it's kind of like the mechanism of why the electrons are going to be going to the oxygen. So I'm not sure if I explained what the oxygen was, but basically what the oxygen is like the final electron acceptor for the whole cellular respiration, right? Like we have the ETC here, for example. The electrons go from the electron carriers, they're going to go down, 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 down eventually, and then the final electron acceptor is going to be oxygen. And then the oxygen collects those electrons and collects some protons from the environment of the mitochondrial matrix, and it produces an H2O. So when we start off here in our first complex of the ETC, we're going to have a high energy electron. Eventually, as it shuttles down, it loses its energy, and then it's collected by oxygen, so O2. All right, and then the concept that you really need to know for the ETC would be the chemiosmotic coupling hypothesis, which basically is saying that because of this proton gradient, it's kind of like a twofold kind of power that it has to land on the oxygen and produce ATP. So the twofold power would be one, the osmotic, osmotic kind of situation, right? There's a lot over here. There's a lot of concentration of protons here. There's not a lot here. So the osmotic gradient is shown, and the protons are going to go down this osmotic gradient, and that power is going to help facilitate the production of ATP. The second, so it's chemiosmotic. The second would be the, oh, sorry. You can add that to your PowerPoint. Would be the electrochemical pressure. So those protons are positively charged, right? So they're going to be kind of repelling each other, and they're also going to be attracted by the electrons shuttling through. So that's another pressure that is used to create ATP. So the osmotic pressure and also the electrochemical pressure. And it is this electrochemical and osmotic pressure that creates ATP. So that's the coupling hypothesis, right? It's a coupling of this pressure with the enzyme in order to make ATP. So the NADHs and the FADHs that come in to donate their electrons, they're going to be, the oxidized forms are going to be produced after ETC, right? Because if they're giving electrons away, they become oxidized. So the products of ETC for a eukaryote, because in eukaryote we have a bit difference in the total number of ATP. In eukaryote, the products of the ETC would be 26 ATP, be 10 NAD pluses, because those NADHs were oxidized, and we get our NAD pluses back, and then 2 FADs, right? Because we got 2 of them from the Krebs cycle, and then we're going to be producing water molecules at the end, as I mentioned before. When the electrons finish with the shuttle, they're collected by the oxygen, and then when you have the protons come back down from their osmotic gradient, those protons, remember that protons is an H+. So the electron plus a proton, like this, is going to combine with the water molecule, and that's why you get H2O at the end. That's why you get 6 molecules of water at the end of ETC, at the end of your cellular respiration. So guys, just remember that the bacteria doesn't have this type of compartmentalization. Like they don't have any organelles. So the ETC is actually just going to be on their cell membrane. Like it's kind of like they're shooting their proton gradients in the environment. Like it's like if they're in water, their proton gradient is in water, and they just they do like that. But they get more ATP than us because their NADHs are all present, right? They don't have any NADHs that are in the cytoplasm that have to like do that whole glycerol phosphate shuttle. Right. So the last thing, that probably the thing that you're going to find the most from ETC, that I would say, would be uncoupling. So here in the chemiosmotic coupling hypothesis, we say that these two pressures, the osmotic pressure and the electrochemical pressure, is coupled with the enzyme that produces ATP. However, you're going to have something called uncoupling, which is when your these two pressures are not coupled with an enzyme. And as we mentioned before, if you go from a high energy state to a low energy state, and there's nothing to kind of like capture that energy is just released as heat. So whenever you have uncoupling, what's going to happen is you're going to have heat released. And for that reason, we mostly find uncoupling in something called brown fat, and brown fat is found in babies, mostly in babies. I think adults have like brown fat, but like it decreases as they age. So babies, they have a lot of brown fat, and that's good because it needs to keep the baby warm. And so uncoupling happens in the brown fat, and just to keep the baby's temperature in homeostatic control, right? And then just the last bit of information is that cyanide is going to be interacting with, the reason why cyanide sucks is because cyanide kind of doesn't let the electrons pass and combine to the oxygen molecule, and so you don't get ATP. So the reason why you die from cyanide is because your ATP is just like depleted, and then you just die, because you need ATP to like have your brain function, for your heart to pump. So it's just a bad situation with cyanide because it just blocks off this last complex. So let's get into the complex, the mechanism of ETC. So here we have the complexes. These complexes are going to be the ones where the electrons are going to be traveling through. So here I have depicted an NADH. The NADH is going to be donating its, or it's going to be oxidizing itself or reducing this here compound. And then this compound / complex is going to take those electrons, is going to pass it on to this, is what's called a coenzyme Q. This carrier is going to then move the electrons and shuttle them to this complex. This complex is going to go to this complex, and etc. until we get to the into the water molecule at the end, right? The water molecule, sorry, the oxygen group is the last electron acceptor. Okay, as here I have depicted, I have the cristae membrane, and these three big complexes are embedded in this membrane. Like they're going to be stuck in there. These two right here that are like kind of between them, they're free floating. And so these embedded proteins or these embedded complexes are going to be staying there, and then coenzyme Q or cytochrome C are just going to be shuttling between these guys back and forth, back and forth. Okay, so how do we name? So that's basically all you need to know for ETC. That NADH sends electrons here. FADH is going to shoot its electrons here. Remember, it's not in the same spot. That's why FADH produces less ATP than NADHs. The electrons are going to move down in this direction. That negative charge from the electrons are going to attract protons. The protons get popped out, and you get your proton gradient. So let's go name these complexes. So it's, again, for biochemistry, I really love that science because they're just so convenient. They're like, okay, let's not make things complicated. So check this out. So this complex here that is going to basically get the electrons from excuse me, that's going to get its electrons from NADH. It's going to be called, and also like once it has the electrons, it's also going to be giving the electrons to coenzyme Q. It's aptly named NADH Q oxidoreductase, right? So it's going to oxidize NADH, and then it's going to reduce Q. So that's an awesome name for this complex because it's just like exactly what it does, right? The name is, it's what it does. So that's the first complex, right? Second complex would be this complex here. So FADH2 is not kind of like be moving on its own. It's actually paired up with succinate. So that's why the second complex is called succinate Q oxidoreductase, right? So the succinate is going to be paired up with the FADH. The FADH is going to be donating its electrons to complex 2, which means that or another way to think about it is that complex 2 is oxidizing succinate, and it's going to be giving its electrons to coenzyme Q. Coenzyme Q is going to go down, pick up the electrons, and then go on to its next complex. So it's succinate Q oxidoreductase. Yeah, this is very, very aptly named. For 3, I've been saying that word, but I hope I'm using it correctly. For complex 3, we have, as you can guess, a Q cytochrome C oxidoreductase, right? Because it takes the electrons from coenzyme Q, has it for itself, and then it's going to reduce cytochrome C, right? It's going to give the electrons to cytochrome C. That's why the oxidation reduction was like a really good, it's a really fundamental, like that concept, the oxidation reduction is really fundamental for you to know biochemistry. So if you didn't show up to yesterday's class, I would highly suggest going through the video that I have up on YouTube, and I have it linked in Sakai. And then the last complex right here is going to be taking the electrons from cytochrome C, and it's going to be called an cytochrome C oxidase, right? Because it's going to take the electrons from cytochrome C. And then lastly, it's going to take those electrons, and it's going to reduce O2. The protons that were up here are going to come down, and the electrons and the protons are going to combine with O2 to form water, right? So really quickly, in terms of like the protons that we talked about yesterday, like, okay, like we said that NADH produces 10 protons like per NADH, and FADH2 only produces 6 protons. So why is that? So we see that the complexes actually are going to be the ones that allow the protons to move over into the opposite side of the membrane. The reason being is that the cristae, okay, this membrane here, is impermeable to protons, right? Like it's not going to let protons come through, and that's where we have such a successful gradient, because the protons are not going to randomly just seep back into the matrix. Remember, this is into the intermembrane space, and this would be the matrix, right? I'm not sure we capitalized it, but I felt important. All right, so this is the intermembrane space. This is the matrix, and so the protons that are here in the matrix. Remember, some protons come through back from the ATP synthase, so there's going to be some protons here, and also just from protons from other reactions as well. So any free-floating proton after the NADH's and electrons go into complex 1, complex 1 is going to shuttle 4 protons into the intermembrane space. The electrons from the FADH2 is going to go into complex 2. The quinone doesn't shuttle any protons, rather it's going to take those electrons and then introduce it to this complex 3, and complex 3 is going to shuttle another 4 protons. After that, the electrons go over to complex 4. Remember, we're going from a high energy state for the electrons to a low energy state, and that's why in the complex 2 we don't shuttle 4 protons, because those electrons already lost a lot of energy, and so this complex 2 is only going to shuttle 2 protons. So let's take a look, right? NADH, we start here. We have 4 protons. The electrons come this way. 4 protons. Electrons come this way. That's 2 additional protons. So it's 4 plus 4 plus 4 is 10. The FADH's electrons only go into this complex and then this complex. 4 plus 2 is 6, right? That's why they have a difference, and the reason why they produce different amounts of ATP, simply because the FADH doesn't start off in complex 1. Complex 1, being the if they ever ask you like which complex have the highest energy of electrons, can anyone tell me?
Complex 1.
Yeah, right, because it starts from a high energy. The electrons are high energy once they pop off the NADH, and then they go lower, lower, lower until only complex 1 can pump out 2 protons, right? Not as much as complex 1. And then you have your ATP synthase that's going to be pumping out the hydrogens and combining those hydrogens and those electrons to the final electron acceptor, the oxygen group. Here again, I have just some nitbits that you guys can follow along with. And the last thing, I guess, one of the side notes for ETC is that when you have electrons coupling with oxygen, what can happen is that if you don't have an even number, like let's say we have some 3 electrons, and maybe, like, of course 4 electrons are going to be popping out, right? But let's say only 3 electrons pair with oxygen, or only 2 electrons, or let's just say an odd number of electrons pairs with oxygen, right? Let's say either 3 electrons or 1 electron. Typically just going to be just 1 electron. The oxygen is not going to have a free radical, right? Because oxygens tend to have like what's it called? 4 valence electrons of its own, and then it's going to be pairing up with something else in order to get its full octet, right? But if you have, let's say, 1 electron binding to it, or 3 electrons binding to it, like such, we don't have a complete octet, and these lone pairs aren't even being shared by an orbital. Like these electrons are being shared by an orbital. Oh shit! These electrons are being shared by an orbital. These electrons being shared by an orbital. These ones, and these ones. When you have an odd number of electrons on an atom, it's called a free radical. And so if you don't have an even number of electrons pairing up with their oxygen, then what's going to happen is you're going to create something that's called a reactive oxidative species, aka an ROS, a ROS. So if you have ROS in your what's it called? In your cell, well, they're highly, highly reactive. So they can just come in and bind to DNA, for example. They could come in and bind to something really important. And if you guys, I'm sure Ashton talked about it, but if DNA is damaged, or certain complexes in your and your cell is damaged, what's going to happen is apoptosis is going to be induced, and then it's going to kill the cell, right? The cell would rather kill itself rather than have something like kind of erroneous happen to it, or have something that's bad occurring inside of it. So I'd rather just like die. It's like, okay, this cell kind of sucks. Like I might as well just like get rid of it, right? So that's ROS. That's something that can happen from ETC if you don't have a proper collection of electrons from your oxygen. And then here we have the regulations. Does anyone have any questions for ETC? That's all that you
I
need
have
to
a
know.
quick question.
Yeah.
So how often do you see it as NADH Q oxidoreductase? Just because the other name I learned it was dehydrogenase.
So a dehydrogenase is like basically the same thing. So what a dehydrogenase does, for example, over here, we have malate dehydrogenase. You see over here the DH. So what a dehydrogenase does is that it facilitates a redox reaction, right? So these are double arrows, which means it can go this way and this way, right? So you can do either oxidation and reduction. And so what's it called? What is an oxidoreductase? That's an oxidation reduction. So another way to say it would be an NADH-Q dehydrogenase. That could be another way to say it. But from what I've seen, like when I took professor, like I had to like pull out my my notes from DICE when I took biochem. It's usually oxidoreductase, but I mean, if you find dehydrogenase, that's why it's really important to understand biochemistry rather than just to memorize it, because that'd be the same thing, right? Complex 1 dehydrogenase, yeah, that makes sense because it does both, right? It oxidizes NADH and also reduces coenzyme Q.
Did it show up as both of them on the MCAT?
I don't remember. I really don't. But when we go over, did you guys have anything about ETC happen in your last two full lengths? Does anyone remember if it came out as dehydrogenase? Because I can amend my PowerPoints to put dehydrogenase if that's what the WMC wants it to be called, But these are the notes I got from my biochemistry class.
I was just wondering though. I don't remember. I don't think we had a question.
But I mean, you guys are ready any case, right? Like you guys know that dehydrogenation. Sorry, that I can't even say now. Dehydrogenase is an enzyme that can do both oxidation and reduction. And what is the complex 1 is doing? It's doing the same thing. So to call it NADH dehydrogenation or complex 1 dehydrogenase, well, okay, that makes sense because it does both. Same thing with complex 2 and complex 4. Okay, any questions for ETC? And also, it's just really fun, right? Like as bio majors, like this is something we can appreciate because it's something that it's not like that's not like obscure like physics. Like a lot of the stuff in physics is just like, well, why do I need to know like resistance and all that unless I'm going to go into like into that field? But this is something that's currently happening in our body millions and billions of times over, like every second. ETC happens every single second. Krebs cycle happens every single second. So it's kind of cool to like understand how the body works. And a lot of like dietitians, like people who work with like how to like manage like foods and like ketogenesis. Like if you've probably heard that name, right? But like ketogenesis is also a pathway in our body. So that's interesting, and like you can learn a lot of things from biochemistry. Okay, so yeah, if I had to choose like my favorite what's it called science from all the MCAT, it'd be biochem. That's why I like teaching it, but I was kind of like stuck with orgo. All right, so all right, any questions about anything we discussed so far? And if not, we're going to finish up. We only have we have several slides to go through, and we're going to start with glycogen metabolism. Okay, if no one has any questions, all right. So what glycogen is, it's just a polymer of glucose, right? This could be a depiction of glycogen. It's a all of these like kind of like let's say there's a dot here, there's a dot here, there's a dot here. All these would just be glucoses and a glycogen polymer. So you can have a glycogen or glucose as a chain, or it can also branch out. So a glycogen is a branching polymer. And glycogen, this whole mess here of glucose, it's really just used for storage, right? Like we talked about how the cell likes to be efficient. And so if the cell is trying to be efficient and like, say there's a lot of ATP in that cell, then it's not going to want to break down its glucose, rather it's going to want to store it so it can break it down later if it needs to. And it stores it as glycogen. So glycogen is going to be your main storage for carbohydrates. you can turn a lot of things into glucose. You can turn like galactose into glucose. You can turn fructose into glucose, as we've seen with glycolysis, right? One of the first few steps, we had glucose turn into fructose. In gluconeogenesis, we'll be seeing how fructose is turning into glucose. All right. So yeah, so glycogen, your polymer of glucose, is stored in the liver and in the skeletal muscle. The reason why it's stored in the skeletal muscle, like, might seem arbitrary, but just think about it, like, if you're running, then you're going to, like, it's not like your muscles have huge, vast amounts of like glucose just floating around. Like, that'd be kind of like silly just to have a bunch of glucose like in your cell, just like occupying a lot of space. So rather, it's like compartmentalized, and it's going to be compartmentalized and stored as glycogen in case you need to like run and you need to like have some glucose to make some ATP. The reason why that it's stored in the liver is that in the liver we have a lot of hormones coming into the liver to regulate things like energy. And so if we have glycogen in the liver, if we ever have something, a scenario in which we need to increase or decrease things like blood glucose, like let's say you're hyperglycemic or hypoglycemic, well, then the glycogen in the found in the liver is going to be making sure that we're staying in our homeostasis for blood glucose. So there are two different things. Liver, we're talking about blood homeostasis for glucose, and skeletal muscle, the glycogen, are going to be used for energy. So that's why whenever you're running and you hit something called, like you hit a wall, like for any of my athletes in here, if you like ever train so hard that you just like kind of collapse, or like you just feel super fatigued and you just need to breathe and chill out, that's because of buildup of lactic acid, and also that your glycogen in your muscles has been wasted, right? So you're gonna have to eat a lot of, that's why they say eat like a lot of, drink a lot of electrolytes and things like that, just to replenish, and a lot of like sugary drinks that should replenish the glycogen that you kind of like used up in your activity. That's why biochemistry is cool. Like, it's a lot of cool stuff to randomly note. Yeah, so in your main chain, so remember we talked about how glycogen is branched, it can be branched. So your main chain, most of it is going to be most of it is going to be in a straight chain, and 10% of it is going to be in your branching chain, or involved with branching. When you want a straight chain of glucose, or sorry, of glycogen, then your what's it called? Your glucose molecules are going to be paired up like that, right? If you remember the drawing of a glucose molecule, this is our carbon 1, this is carbon 2, this is 3, and this is 4. So what alpha, can anyone tell me what alpha is? Like, what does that mean? If you guys remember Orgo.
It means that the group on, I think it's, I forgot which carbon it was. It was like this
On
one.
anomeric carbon,
Yeah, that
it's
one was
gonna be
in
hooking
position.
down.
Thanks, Karen. Karen paid attention. Yeah, so as Karen and Kevin combined forces, yes, it's going to be this anomeric carbon over here, carbon 1. So when it says alpha, it's going to be, this carbon's pointing down here, and this is carbon 4. And in carbon 4, we also have the oxygen pointing down. And so they're just going to have your glycosidic linkage, something you might have learned from biology. And it's going to, basically, if we have another, let's say, glucose molecule right here, the fourth carbon is going to combine, like this is number 1, this is 4 on this one. So they're going to combine as such and create a chain. However, when you're talking about a branching glycogen, or whenever you have a branching structure, all you're going to have is the same situation, except that you're going to have the sixth, what's it called, oxygen on your 6th carbon interact. Right, so you'd have something like this is carbon 1, this is carbon 6 right here, and these guys bind for your glycosidic linkage, and then you have a branch. And what's cool about this is that the 4th carbon in this group is still accessible. So you can have more branching this way. Sorry, you have a straight chain this way. This carbon is still accessible, and then you have a carbon this way. Remember, it's alpha 1,4, and then this 6th carbon, combined to another glucose, whatever. And then you have it this way, right? So that's how glycogen works, right? Just that branching. Yeah, biochemistry is fun, dude. All right, so, because I didn't prepare this like little example that I just showed you, but it's intuitive now, right? Because like the alpha 1,4 and the alpha 1,6, well, this is something that can happen, right? You have your branching, and then you have something along those lines keep you going, because your 4 and your 1 of different glucoses are still open. So you can, okay. So glucose and glycogen, they're polar, right? They have just a ton of OHs. And remember, we said that polar things dissolve in polar things, right? So glycogen is going to have a water shell around it. It's going to be surrounded by water, and that's what's known as your water weight. It's like, oh, I just have to lose water weight. Well, just the water that's found on your glycogen. That's something I read in my notes that I from BioChem. Whether that's 100% true, I'm not sure, but that's just, I think it's a cool little fun fact. I'll double check it with Google if that's like what actually water weight is. On the other hand, we have fat, which is like not polar. And if it's not polar, then it's not going to be having that water shell around it. And that's why fat is like kind of like what's it called? It's not really that heavy. Like if you had like a handful of muscle and a handful of fat, like the muscle is going to be super, super heavy. The fat, on the other hand, not really. For the same reason, you have a handful of glycogen and a handful of fat. The glycogen has that water surrounding it, making it heavier. Okay, so that's glycogen metabolism. Here I have some notes again. It's like a gram of glycogen will get you 4 kcal or calories. A gram of protein will get you 4 calories, and a gram of fat is going to get you 9 calories and give you more energy. And that's why fats are usually used for storage of energy, and that's why they're really good for storing energy. And that's why we find fatty foods so yummy, is because our bodies, like back in the day when we didn't know when our next meal was going to be coming from, we just needed calories. We just need something to power ourselves, right? And fats, they have a potential to drop like a glucose molecule can like drop 2 acetyl-CoAs, and then you can get the ATP out of that, right? A fat, like look at all these carbons, right? Like it can be like 18 carbons. That means there's 9 acetyl-CoA that can be used. Like that's a lot of energy. And that's why whenever we eat something that's like fried or anything like that, we find it so yummy, because our body's just like, oh wait, that's a good energy source, so I'm gonna make it yummy for you. Okay, like in our heads. All right. Any questions about glycogen metabolism? Okay, if not, the mechanism for it, it's right here. I'm going to go through all the theory for the remaining pathways first before I get into the mechanism. You guys can just go over the mechanism on your own. The structures are drawn here. Just remember to recognize enzymes like hexokinase. Oh, so with Julia yesterday, she asked me like, oh, what does the hexo mean in hexokinase? Like, does it tell you that it's going to phosphorylate it on the 6th carbon? And I was like, okay, I'll look it up. And I looked it up, and it just means that you're phosphorylating something that is a 6 carbon structure. So, but what we've seen, as a trend, that the name, but I don't want to say that, oh, like there's a trend and then have someone trip up, but just just know that if there is like a prefix for a number before something, that just tells you like the structure of the molecule that you're working with. Okay, so we'll go over this last, like the mechanisms we'll go over last, simply because they're just really, really tiny, and we can actually, you know what? There's a lot of information also joined with the mechanism, so we'll just go over the mechanisms first. Who cares? All right, so here in glycogenesis, if anyone knows, genesis just means starting. So we're going to be making, what's it called? We'll be making glycogen. Glycogenesis, we'll be making glycogen. So here we have our glucose. It's doing its thing. Hexokinase is a enzyme that puts a phosphate group, here kinase, on a 6 carbon molecule. It's going to funnily put it, is that a word? Is going to put it on a 6th carbon here. We're going to have glucose 6-phosphate, and then we're like, that's the first step for our glycolysis step, right? That's the first thing that we do for glycolysis. But as I mentioned in yesterday's class, this glucose 6-phosphate is not only involved with glycolysis, it can also go into glycogenesis. Okay, so next we're going to have our phosphoglucose. Remember, all your enzymes are aptly named. It's awesome. Phosphoglucose. Remember, you have a phosphate on a glucose. Mutase. What the mutase is going to do is just going to make a isomer. Right? So instead of having the the phosphate on the 6th carbon. We're going to have our phosphate on the 1st carbon. After that, we're going to have a UDP transferase enzyme transfer a phosphate over to our our glucose 1-phosphate. So what a UTP is, just we have ATP, adenine triphosphate, we have guanine triphosphate. This is just the urine version of it, uracil triphosphate. So you have a uracil triphosphate, so a uracil with 3 phosphate groups. It's just going to combine one of those phosphates with our glucose 1-phosphate, and then it's going to ditch 2 phosphates, right? So like if this was our uracil triphosphate, it's going to ditch these 2, and it's going to come in and bind to this phosphate over here. And that's why we have this structure right here with the 2 phosphates and the uracil still hanging on, and aptly named UDP glucose, right? Very, very awesome naming here. Then we have UDP glucose combining it with glycogen. So something I failed to mention over here is that a glucose on a glycogen, like a single glucose, is called a residue. So a chain of glycogen would be just a bunch of residues of glucose. So over here, we have UDP glucose. We have glycogen with n number of residues. Like we don't know how, let's just say an arbitrary number of residues, right? Residues mean just 1 glucose. And then we're gonna look at this enzyme. Glycogen synthase, like who does that? Like that's just too easy. So glycogen synthase is going to produce glycogen. So this glucose is going to come in and bind, make glycogen with an n+1 residue, right? Because we just added a glucose, so it's n+1 residue. And then the 2 phosphates that were on the glucose is going to be released, and then it's going to be released as uracil diphosphate. So that's the mechanism for glycogenesis. It's stimulated by insulin, right? Because remember, in insulin, we want things to be going out from the bloodstream, right? Like it's going in, as Pepper said. So we have oh shit, I wonder why it does that. Okay, we have this right here. We have our cells. We have glucose in our bloodstream. If we shoot our glucose in, and let's say, for example, our cell doesn't really need that, it doesn't need that glucose per se. We just want to make sure that it gets out of the bloodstream. Then what it can do is just, it can make, what's it called? Glycogen, right? It's just like, okay, we have enough. We don't necessarily need it. So let's just, make glycogen out of it. So this process of glycogenesis is stimulated by insulin. Whenever you're adding a new glucose group, you're going to be adding it to the non-reducing end of your glycogen. And as we mentioned before, glycogen is branched. So if like this would be this for glycogen synthase is when you're attaching an alpha-1,4. But if you're using a branching enzyme to make a branch, you're going to be using a glycogen synthase from the alpha amylase family. Something that's kind of like low yield, just like kind of to make things full circle. Right? We talked about branching, got to have the enzyme, the branching enzyme as well. Right, any questions about I can never say the word glycogenesis? Okay, if not, then let's move on to glycogen lysis. So again, very awesomely named, glycogen, and then lysis means to cut. So we're going to start with our glycogen.
Wait,
Jesus.
I have a quick question.
Yeah.
So in previous slide, you mentioned something about non-reducing end. Can you remind what it is? Like, do we have to know that? Like how it looks like? Or no?
Yeah, you do, but I haven't gone over that yet. So
It's okay. Sorry, it's okay.
I mean, I will have it up as a recorded lecture. I'm still going to. So again, it's like there was so much to cover with organic chemistry. I want to take my time with Orgo because a lot of people don't like Orgo. I think now that I'm teaching Orgo, I guess I have a good appreciation of it. But Orgo, people don't like Orgo, so like I try to take my time with it. But basically, I'm going to be sending every single chapter that the TPR books have as a recorded lecture if we don't finish it in class. So even okay, all right.
Thank you.
Yep, no problem. So yes, we'll discuss what a non-reducing end is soon in a recorded lecture. But again, you guys can don't wait for us to present the information for you guys to go ahead and tackle. Always make sure this class is really just to facilitate the process. Make sure that you're studying on your own as well. You shouldn't only be studying Fridays and Saturdays. When the spring starts, you should be studying every single day, a little bit, at least cars, I would say. So All right, so here we have the glycogen lysis. Remember, lysis means to cut. So we have our glycogen. Let's just say it's 1 to 4, which means that's just a straight chain. And then we have something called the glycogen phosphorylase. Remember, here in the glycogen genesis, we had the phosphate group on our glucose. So we have to do the same thing with the glycogenolysis. So if we want to pluck off a glucose from our glycogen chain, we're just going to have to add a phosphate to it. And remember, phosphorylase means that it's going to be an inorganic phosphate, and HPO4 fits that bill. It's an inorganic phosphate. And then we're going to make our glucose 1-phosphate, and we're going to have a glycogen that's -1 residue. Remember, residue means number of glucose. And then next, we're going to have our glucose 1-phosphate. We're going to use PG mutase. PG mutase was also found in this process right here. It was right here. PG mutase is phosphoglucomutase. So we're going to turn our G1P into glucose 6-phosphate. That's what we find, glucose 6-phosphate. And then we're just going to use glucose 6-phosphatase, super awesomely named, again, glucose 6-phosphatase, just to produce glucose. Okay, and this is, if this process was stimulated by, what's it called, insulin, this one's going to be stimulated by glucagon. So I'm curious, what was the analogy or the way to, okay, I accidentally paused the recording. I was trying to go to chat. Oops. Okay, so insulin in, insulin in and glucagon gone, meaning we need to start breaking down stored glycogen. Yeah. Yep. So just glucagon gone. So for so glucagon is going to stimulate glycogenolysis, right? Because we need glucose. So like, okay, let's get it going. Let's get some glucose off from our glycogen. And then for epinephrine, another name for epinephrine is called adrenaline. So I don't know if you guys knew that, but I think the British term for epinephrine is adrenaline. And what happens if you were to shoot yourself with adrenaline? Can anyone tell me? Someone who drinks Monster on the daily, can anyone tell me what happens when you have too much adrenaline? No. Basically, you get super hyped up and you can do whatever you want, right? You just have so much energy that you can like run a marathon. So again, if you have epinephrine in your system, you're going to need some glucose, right, to get some ATP to get yourself moving. So epinephrine really just it makes sure that you get some glucose from your glycogen. So it stimulates this process. Okay, next we have the von Gerk's disease. So I'm pretty sure I'm saying that wrong. Von sounds like it's German, so von Gerk disease. My best attempt. What happens with von Gerk disease is that you don't have this enzyme called the glucose 6-phosphatase. It's this last one here. And if you don't have this enzyme working, then you can't get glucose from glycogen, which means that you're just going to be super, super low in your blood sugar, right? Because if you can't get it out right here, you can't get any glucose into your bloodstream, and it sucks, right? That's von Gerk's disease. And of course, if we have a branching enzyme, we're going to have a debranching enzyme. All right, any questions about glycogen metabolism? If not, all right, let's continue onward. We have the pentose phosphate pathway, also named the PPP. It's going to be in the cell cytoplasm. So what the PPP does is that it gives you something that Matt talked about yesterday. It's going to give you the NADPH. That's also going to provide for you ribose 5-phosphate. And additionally, depending on the mode that you use, is going to give you glycolytic intermediates. So what the pentose phosphate pathway does is that it takes the glucose 6-phosphate. Remember this glucose that we start off with, the first step in hexokinase. You take this glucose. Remember, this glucose 6-phosphate has a lot of things that it can do, right? Not only just glycolysis. For example, in the PPP, glucose 6-phosphate is taken. It's super duper oxidized. Remember, you're oxidizing something. There's got to be something that captures it. The NADP plus is going to capture it, making NADPH. The structure that you had once before, glucose 6-phosphate, that backbone is going like more. You're going to have a carbon dioxide form, meaning you're going to have a decrease of 1 carbon, and you're going to be making ribose 5-phosphate. That looks pretty familiar, right? Or ribose sugar. Can anyone tell me what a ribose sugar is useful for?
Synthesis of nucleotides and nucleic acids.
Yep. So ribose is going to be what you're made out of if you're a DNA molecule or you're an RNA molecule, right? So it's a really important pathway. Also makes glycolytic intermediates. So NAD, so okay, what's the difference between NADPH and NADH? Well, just think that NADPH is like Superman, you know, he's like super strong. NADH, like NADH is your regular schmo. NADPH is Superman, he's got, you know, super duper awesome reducing powers. And for that reason, NADPH, rather than acting as an electron carrier that's going to go to ETC to produce ATP, is going to be more focused on reducing stuff. So NADPH is going to be, you know, a popular and very specific reducing agent in the fatty acid synthesis. Is going to be a pathway we talk about tomorrow. It's going to be one of the last pathways that we do. And it's also such a good reducing agent that is going to help with the elimination of those reactive oxygen species, right? Those reactive oxygen species is going to help fight those by reducing something like called glutathione. It's going to, like, you know, there's something called antioxidants, right? What's an antioxidant? Something that kind of reduces oxidative species. That's what an antioxidant does, you know. But that what the NADPH is going to just recharge your antioxidant, just so it can keep going and keep eliminating these ROSs. Okay, so yeah, the pentose phosphate pathway is also known as the hexose monophosphate shunt. Why? Because the what is shunted from the glycolysis pathway? Well, glucose 6-phosphate, right? Glucose 6-phosphate was originally in the glycolysis pathway, but it became shunted and it's entering the PPP pathway. Right? So that's what's called a hexose did I spell that wrong? Right. Hexose monophosphate pathway, because the hexose with a single phosphate on it. Right? Everything in biochemistry very aptly named. I guess like biochemistry would be like the psychsoc of like what's it called, of science, right? Everything has the proper name. And then just something to note, ATP is not used or consumed or produced in the PPP. Okay, any questions so far about the this pathway?
So just to confirm, whenever they refer to like a shunt, they mean that's bypassing something?
I'm not sure how many shunts you'll find in biochem. Like, this is the only shunt that I ever found. But I would say in this case, the shunt is that it's just taking something that would belonged originally to a different pathway. So it's like shunting. It's like kind of like if you think of a tube like this, and then the shunts would just be like, okay, like we put the a lever down, and so the thing that was supposed to be going down just like went this way instead.
Okay, because I remember it was like in bio as well.
What was the thing?
It was the ductus venosus.
The ductus venosus. Is that?
It's like a fetal shunt. Like, don't worry about it. I'm pretty sure it might be like 2.
Sounds like something regarding the, because I know what a duct is, like the you know, I mean, maybe I'm thinking of a sphincter. No, I'm thinking of a sphincter. Yeah, cool. But yeah, I mean, shunt in this case right now is just saying that, okay, it's originally supposed to be for glycolysis, the G6P, but it just got shunted into a different pathway. In terms of like where shunt is used in terms of biology, I can look that up for you guys for sure. All right. So in the PPP pathway, the pathway, we have 2 separate pathways. We have the oxidative pathway and we have the non-oxidative pathway. And the oxidative pathway, something that's irreversible, we're going to be producing NADPH and our ribose 5-phosphate. In the non-oxidative pathway, we are going to, oh nice, and glyoxylate shunt. Yeah. I'm kind of freaking myself out. I didn't know that biochemistry was this fun. Yeah. All right, so let me see here. All right, so then we have a non-oxidative pathway in which we, instead of making the NADPH or the ribose 5-phosphate, we're going to be working with glycolytic intermediates. So if we're talking about glycolytic intermediates, there's a lot of carbohydrates involved with glycolysis, right? So we're just going to be working with different carbon structures in this non-oxidative pathway, and all the reactions that happen in the non-oxidative pathway are reversible. Oxidative, irreversible, non-oxidative, reversible. Okay, so before we get into the modes, I'm going to show you guys the pathways. Oxidative. Okay, so here we have the oxidative branch. Remember, we have the homeboy, the glucose 6-phosphate. We're going to get it first by phosphorylating the glucose with hexokinase. and then we're going to get our glucose 6-phosphate. Next, we're going to have the usual suspect, the dehydrogenase, that's going to be used in our redox reaction. We have glucose 6-phosphate dehydrogenase. You guys should get used to these abbreviations by now. Instead of using NAD+, we're going to be using NADP+, right? Because I believe, no, there's actually another pathway. This is one of the only pathways that produces NADPH. I think there's another one that also produces NADPH, but whether or not you guys need to know that pathway, I'm not sure. We'll get to it if it pops up on our phone. It's like a very small pathway. It's like very kind of like, I'm not even sure if I can find it in my notes, but yeah. So we oxidize our glucose. As we can see here, this was originally a what's it called? An alcohol group. Now it's a carbonyl group, right? The hydrogen was removed. It's been oxidized. We produce our NADPH. Again, very aptly named. Look at this name: 6-phosphoglucanolactone. Do you guys remember what a lactone is? Where's the lactone in the structure?
I think it's that like ring-like structure, like like with that like double bond, like to the right, and that like ether type piece. Yeah.
This is your lactone. If we're talking about a lactam, we got this, right? You have an amide, kind of like an amide ring. And here we have an ester ring. Okay. So again, very aptly named. It's really cool how they intersect, right? Organic chemistry and biochemistry. So we have a 6-phosphoglucanolactone. And then we have a lactonase, okay, awesomely named enzyme again, lactonase. And it's just going to open up this lactone, and then we're going to have our 6-phosphogluconate. Right. Next, we're going to use a 6-phosphogluconate dehydrogenase. So don't get these two enzymes confused. I know they look similar. This one's a glucose 6-phosphate dehydrogenase. This one is a 6-phosphogluconate dehydrogenase. So the P is just here, it was at the end, here it's in the middle. I want to get another NADPH. We're going to have a brief structure form here, and then we're just going to extract a CO2. Let me just. If we have a 6-carbon group and a CO2 was released from that 6-carbon group, then what we're going to be left with is a 5-carbon group, and that's going to be our ribulose 5-phosphate. It's called a ribulose. That UL is there is because we have a ketone. If we were to oxidize it, if we were to reduce this ketone and instead make an aldehyde, we get the classically named ribose instead of ribulose. So we're just going to use a phosphopentoisomerase, again, very aptly named phosphopentose, so 5-sugar isomer, right? Just the carbonyl is like not in the second carbon more, now it's in the first carbon. We have ribose 5-phosphate. So this is the oxidative mech, the pathway, right? This first, what's it called? Reaction is irreversible. This one is also irreversible, and this one happens to be reversible. These are the most important parts, right? When you're producing your NADPH. There we only have one point of regulation here. It'd be the first reaction. Just the products of the first reaction is going to negatively inhibit it. As we mentioned before, the NADPH can be used to help rejuvenate antioxidants such as glutathione. And if you can't make NADPH, for example, like if you don't have a properly functioning glucose 6-phosphate or a properly functioning 6-phosphogluconate dehydrogenase, this is a deficiency of glucose 6-phosphate dehydrogenase, or the thing that produces NADPH. It's called phobbism. And if you don't have that, then what's going to happen is those reactive species is going to really just tear up your cell, right? Those things are just really, really reactive. They're going to tear up your cell, and it's not going to be good. Okay. That's why NADPH is really important, right? It's a reducing guy. It's not really a person who donates its electrons to ETC. Okay, the last pathway for today, I'm pretty sure we're going to finish early today, which is a new one for us. But in the non-oxidative reaction, we have the combination of different carbonyl sugars, right? So we have either a ketone for ketose sugars or aldehyde for an aldol sugar. So okay, what's this? It's kind of like a puzzle, I would say, the PPP, non-oxidative mech. Remember, they're all reversible. So we either have we have either the 10-carbon pathway or we have the 9-carbon pathway. Here I have an example of the 9-carbon pathway. Let's go into the ground rules first. So every time you use a trans-ketolase, you're going to be moving 2 carbons. If you have a trans-aldolase, you're going to be moving 3 carbons, okay? And what you need to note is that the keto sugar, so this is a ketone right here. If you have a CHO, that's an aldehyde, right? CHO just looks like this, right? That's an aldehyde. If you have a ketose sugar, the ketose is always going to be donating its carbons to the aldehyde. And for that reason, your ketone sugar has to be bigger than the aldehyde, always. Like the ketone is always going to be the one that's the bigger of the two, and the ketone is going to be the one that's donating the aldehyde. Okay, so how does this all work, right? It's just it's kind of like a puzzle, right? So if you have fructose 6-phosphate, right? Fructose is a 6-carbon sugar with a ketone. And we had G3P, aka glacier aldehyde 3-phosphate. That's something that we remember from the glycolysis pathway. Here we have the ketone here. Here we have the aldehyde here. The ketone is going to give it, depending on the enzyme. Here we have a trans-ketolase. It's going to be giving it 2 carbons, right? So if you move 2 carbons to something that has 3 carbons, then what's 2 minus 6? That's 4. So then at the end, we're going to be having an erythrose 4P that came from the fructose, and then 2 carbons added to this 3-carbon group. We're going to have a 5-carbon group, and then you have a xylose 5-phosphate. What's important to note is that the phosphate is always going to be at the end, right? So it's always going to be like, if it's a xylose, it's 5-carbon sugar, it's going to be 5P. Erythrose is a 4-carbon sugar, it's going to be a 4P. So here, the ketone gives the carbons here. And then when the ketone gives its carbons, it turns into an aldehyde. And the aldehyde turns into a ketone. I should probably write that down. 2. Ketone turns into the aldehyde. And vice versa. Oh. So if you didn't download the PowerPoint already, I would just say I'm going to upload the new version. I made some corrections. Oh, I don't think it's going to crash again. Yep, crashed again. But okay, so this would be the 9 pathway. We have the 10-carbon pathway. It'd just be the same process, right? Just you're using a trans-ketolase, you're moving 2 carbons. You're using a trans-aldolase, you're moving 3 carbons. Just keep these rules in mind. And for example, here we have a xylose, which is 5, a ribose, which is 5. And then if you're moving only 2 carbons, right, trans-ketolase, you're going to have something that has 7 carbons, like this said. I don't even know how to say the name, but I just said. And then you're going to have something that has 3 carbons. It would be like your glucose 3, your glacier aldehyde 3-phosphate. So as you mentioned before, these intermediates can go back into the glycolysis thing, right? So fructose 6-phosphate, that's something that we found in glycolysis, right? Something after we isomerized our glucose, we made a fructose. So fructose from the PPP can go back into glycolysis, and the glacier aldehyde 3-phosphate, something that's in the second part of glycolysis, can go also into the back into the glycolysis pathway. Okay, so the last thing for today, and then we're done. There's different modes of the PPP pathway that you can have. So the first mode, M1, is when you only want to produce ribose 5-phosphate. Okay, well, if we only want to produce ribose 5-phosphate and not NADPH or ribose 5-phosphate, if we like, okay, maybe we say that. We have our first mode in which we produce only ribose 5-phosphate. So if that's the only thing that you want, like you don't want NADPH and you're trying to use the PPP pathway, then all you're going to use is the non-oxidative reaction, right? Like in this non-oxidative reaction, no NADPH is formed, right? So that would be M1. M1 only want to produce 5-phosphates and then use the non-oxidative pathway. If you want to produce both NADPH and ribose 5-phosphate, well, you're just going to have to use the oxidative pathway and only use that one, right? Because you have a ribose 5-phosphate here, and you also get 2 NADPHs. However, if you only want NADPH, then what we're going to have to use is you're going to have to use both, right? So you're going to have to use the oxidative pathway to get your NADPH. At the end, you get a ribose 5-phosphate. Okay, how can you get rid of that? Well, you introduce that ribose 5-phosphate into the non-oxidative mech. and then that ribose 5-phosphate is going to be used up in some sort of reaction. Okay, any questions about the PPP? If not, you guys are free to go, and I'll be working on putting up that second video with gluconeogenesis and the Cori cycle and fermentation. And then tomorrow we're going to have our last pathways, and then we're going to dive into enzymes and amino acids, and then that'll basically be it for biochemistry. That's the main things you need to know. You need to know amino acids, enzymes, and pathways. That's all, really.
Thank you. Appreciate it.
No problem, man. See you guys tomorrow.
Thank you.
Yo.
I just got, because I was in the shadow of things, so I don't know how I was credit for.
Oh, wait. Let me stop the recording.